Ooh.
The topic of today class is medical genetics. The topic of today lecture is methods of human genetics. The man is unfavorable object for a genetic study because it is very difficult to study human inheritance and variation. We can't use breeding methods for studying of inheritance, where people mostly have a small number of offspring and the long time for next generation is characterized for people too. So all these features do studying of human inheritance very difficult. The people have a large number of chromosomes and large number of genes. In human genotype, in human genome, we can find near of 40,000 genes. So the scientists used for studying of human inheritance and variation were some methods, and these methods has a name of methods of medical genetics or methods of human genetics. The first and one of the most early method of human genetics is pedigree analysis. A pedigree is a diagram of family relationships in which symbols are used for represent people and lines are used to represent genetic relationships. These diagrams make it easier to visualize the relationships within the families, particularly large extended families. Pedigree are also often used to determine the mode of inheritance of the genetic diseases. So, the pedigree shows the transferring of characteristics from generation to generation, and we can determine the mode of inheritance. We can determine the level of this inheritance and others. For pedigree analysis, we can use these symbols. So we can use a pedigree analysis to study different modes of inheritance. For best studying of mode of inheritance, we should know about more as free generation, about characteristics in more and free and in more as free generation. The person which pedigree we study has we call proband, and with help of pedigree we can study the inheritance of a single gene human characteristics. So we can't use this method for diagnosis of for studying of chromosomal disorders. This method studies the single gene inheritance. There are 6 types of inheritance. And we can find these types with help of pedigree analysis. The first is autosomal dominant inheritance. The major characteristics of this type of inheritance are the characteristics manifest in heterozygous stage. So we can find characteristics in heterozygous and dominant homozygous. The gene located in autosomes, so we can find these characteristics equally in males and females. Vertical family history may be seen, and male to male transmission is possible. So the examples of autosomal dominant human disorders, these are Marfan syndrome, Huntington disease. The autosomal dominant human characteristics, these are dark hair, black eyes, the using of right hand. So all these characteristics are autosomal dominant. So students, here you can see the pedigree of autosomal dominant type of inheritance. So you can see which the characteristic presence in each generation. The person with characteristics is possible if both or one parent have this characteristic, and the vertical type of inheritance is characterized. And we can find these characteristics equally in male and female. The second type of inheritance is autosomal recessive inheritance. The gene is located in autosome, so we can find these characteristics equally in male and females. In female, the child with characteristics can be born in parents without characteristics. Pedigree may show several SIPs and cousin affected in same generation. The horizontal transmission is indicated. Very often, the people with characteristics are born in consanguineous marriages. The example of autosomal recessive human disorders are sickle cell anemia, phenylketonuria, albinism, cystic fibrosis, homocystinuria, and others. So you can see which the characteristics is not present in each generation and equally in male and female. The next type of inheritance is X-linked recessive inheritance. So when gene is present in X chromosome, the gene is recessive. Mostly the males are affected, but the transmission is through heterozygous females. So we call these females carriers. No male-to-male transmission, and all daughters of affected fathers are carriers. The examples of X-linked recessive type of inheritance, this is Duchenne's muscular dystrophy, hemophilia, color blindness. So you can see which mostly we can find the X-linked diseases in males, but the transmission directly from male to male is absent. So if the male is sick, so the next sick male will be maybe son of daughter of this male. So for this type of inheritance, the skipping of generation is characterized. The next type of inheritance The next type of inheritance is X-linked dominant inheritance. We can find these characteristics in males and females, and in females more often. Where characteristic is present in each generation, but the female with characteristic transfer these characteristics to daughters and sons, but the male transfers only to daughters. So, X-linked dominant characteristic, the male transfer these characteristics to sons and to daughters. We can find this characteristic both in male and female. But the male transfer this characteristic with X chromosome only for all daughters, but all sons will be without this characteristic. Next type of inheritance is Y-linked inheritance. This type of inheritance has other name, holandric inheritance. This is inheritance of genes which are present in Y chromosome. Only male are affected. The characteristics we can find in each generation. The affected males directly transfer this disorder to their sons, and sons transfer to their sons and to other sons. The example of Y-linked inheritance, this is hypertrichosis of airlops. The next and last type of inheritance is mitochondrial inheritance. This is inheritance of genes which present in mitochondria. So we can see which the male and female can be affected. But the affected female transfer these characteristics to all sons and daughters, and affected male doesn't transfer this characteristic to his children, because during fertilization from male only genetic material of nucleus take part. So students, the pedigree analysis played a very important role in development of medical genetics, because each helped and each helps in finding of mode of inheritance of different human diseases and different human characteristics. The next group of methods of medical genetics, these are cytogenetic method. Cytogenetic methods. This is group of methods which based on studying of chromosomes. This method includes karyotype analysis, examination of bark body, chromosome binding, and molecular cytogenetic. The chromosomes, these are structures of nucleus. These are microscopic filamentous structures which contain an individual's genetic material. The chromosome consists of chromatin, and the chromatin is complex of DNA and proteins. The chromosomes carries genes, and the genes are arranged in a chromosome in linear order. In human somatic cells, we can find 46 chromosomes, which are arranged into 23 pairs. These 46 chromosomes carries 40,000 genes. We can tell which these 46 chromosome we arranged into 23 pairs, and where one set of chromosomes, 23 chromosomes, we inherit from mother, and other 23 chromosomes we inherit from father. Chromosome analysis or karyotype analysis, this is a starting of number and general structure of all 46 chromosomes, and each has name karyotyping or karyotype analysis. So this method studies number and structure of chromosomes. But you should remember this method doesn't studies the single gene mutations, this as sickle cell anemia. So we can study number and structure of chromosome by karyotype analysis, but we can study the teeny small changes of chromosomes as gene mutations. The all chromosome abnormalities we can divide into groups. These are numerical chromosomal abnormalities. The reason of these abnormalities, this is changes in number of chromosomes. So the reason of these abnormalities is aneuploidy or polyploidy. Mostly in people we can find trisomy. This is 47 chromosomes. More rare, we can find 45 chromosomes. This is monosomy. The examples of trisomy in people, this is Down syndrome, Edwards syndrome, Patau syndrome, Klinefelter syndrome, trisomy X, and Jacob syndrome. The example of monosomy is Turner syndrome. The second group of chromosome abnormalities, this is a structural chromosome abnormality. These are changes in structure of chromosomes. There are four main types of changes in structure of chromosomes. These are translocation, inversion, duplication, and deletion. This rearrangement may or may not result in obvious health problems. This depends on whether the structural problem ultimately results in a net gain or loss of chromosome material. If a chromosome material is simply in rearranged fashion, but all genetic information is present, so in this way, in this case, the person doesn't have any symptoms, and we call this condition balanced rearrangement or balanced mutation. So if we tell about first group, numerical chromosomal abnormalities, very often they cause very severe abnormalities in the body. And very often the people with these chromosomal abnormalities die in early stages. Structural chromosomal abnormalities cause not so severe abnormalities of the body, So, karyotype analysis involves visualization of chromosomes under microscope. For karyotype analysis, we can use the somatic cells of our body. Uh, the best cells for karyotype analysis, these are cells of bone marrow, because these cells divides very quickly. But it is very difficult to remove these cells from our body. So very often for karyotype analysis, we use the lymphocytes from our blood. But the lymphocytes, these are cells which present in a GO period of cell cycle. So these are high differentiated cells, and they don't divide. So for stimulation of mitosis of lymphocytes, we use mitogens. These are drugs which induced mitosis. Very often we use phytohemagglutinin as mitogen. After this, on metaphase stage, we arrest the mitosis by using of colchicine or colchicine, which this is drug which disrupt the mitotic spindle and arrest the cell in metaphase. After this, we use hypotonic solution. Each causes the swelling of nuclei and destroying of the cell. After this, we stain chromosomes and stain with Giemsa dye and after Giemsa dye. And after this, we see these chromosomes under microscope. But we see under microscope the mixed chromosomes, and it is very difficult to use these chromosomes for diagnosis of human disorders. So the next work is the chromosome should be photographed, cut out, and assigned into appropriate chromosome number, or they can be digitally imaged using a computer. The chromosomes can be divided into 7 groups from A to G based on descending order of the size and position of centromere. The standard nomenclature of describing a karyotype is based on the International System for Human Cytogenetic Nomenclature. So this procedure of karyotype analysis. So you can see the main step of this analysis. So as a result of these steps, you can see this picture, you can see the lymphocytes, and you can see the chromosomes which are released from these lymphocytes. And you understand this picture is we can't use for diagnosis of different chromosomal disorders. So the special computer program do the human karyotype. This artificial scheme of human karyotype has name I-diagram. In this I-diagram, the chromosomes are arranged from largest to smallest, but the last pair of chromosomes is sex chromosomes. So you see which in somatic cells of normal people, there are the 46 chromosomes, 42 autosomes, and 2 sex chromosomes. So this is normal male and female human karyotypes. The second method of cytogenetic methods, this is examination of Barr body. Where in previous century, the scientists found which in most number of cells of female body, the small black spot is present. This spot was called by Barr body, and now we know which in women where second X chromosome present in inactivated condition, in condition of heterochromatin, and we can see this second X chromosome as Barr body, dark black spot in the nucleus. So you can see the Barr body in cells of buccal cavity. This is dark spot, or we can see the second photo shows to you the Barr body in human white blood cells. So students, if we tell about karyotype analysis, the karyotype analysis, this is very long and expensive method because we need the special equipment, we need many chemicals for providing of this method, and we need time for culturing of the cells. The examination of Barr body is a very easy method. For this method, we should need the special stain, we should need a simple large microscope, and we need glass. And during of 15 minutes, with help of this equipment, you can detect the gender of your patient. So examination of Barr body, it is very cheap, very rapid, very easy method, but it shows us only the changes in number of X chromosomes. So with help of examination of Barr body, we can diagnose this the Klinefelter syndrome, Turner syndrome, trisomy X. But we can do this very, very quickly. The next groups of cytogenetic methods, these are chromosome bending and molecular cytogenetics. Chromosome bending, these are different staining methods which can be utilized to identify individual chromosomes. There are 4 main types. This is G bending, Q bending, R bending, and C bending. Very often, the G bending is used where chromosomes are treated with trypsin to denaturate their protein content, and then stained with DNA binding due known as Giemsa, which gives each chromosome a characteristics and reproducible pattern of large and dark bands. Q bending gives a bending pattern similar to that obtained with Giemsa and requires examination of chromosome with an ultraviolet fluorescent microscope. bending. In this technique, the chromosomes are heat denaturated before staining with Giemsa, yielding large and dark band patterns, which are reverse of those obtained using of um, G bending. And C bending, the chromosomes are pretreated with acid prior to G bending. The centromeres and other heterochromatic regions containing highly repeated DNA are preferentially stained. So, four types of bending. So, in G bending, we use Giemsa stain, and each gives chromosomes a stripped appearance because each stains the regions of DNA which are rich in adenine and thymine base pairs. By Q bending, um, we use alkylating agent or quinacrine dihydrochloride. Q bands are alternating bright and full fluorescent bands. Bright bands, regions of DNA rich in adenine and thymine. Dull bands, regions with guanine and cytosine. This bending is used for identifying of human chromosome Y and various polymorphism. Binding. This is bending which has a reverse pattern of G bands. Pretreating cells with hot salt solution causes denaturation of DNA rich in adenine and thymine. After this, these chromosomes stain with Giemsa stain. This bending is used for analyzing of structure of chromosome ends. C bending stains the constitutive heterochromatin, which usually lies near of centromere. where molecular cytogenetic includes fluorescence in situ hybridization, which has name FISH. This is molecular cytogenetic techniques which used fluorescent probes which bind to only those parts of the chromosome which a high degree of sequence complementary. It is a technique used to detect the presence or absence and location of specific gene sequences in chromosomes. FISH is a process which vividly paints chromosome or portion of chromosome with fluorescent molecules. Now we can use this method for diagnosis of different chromosomal disorders and diagnosis of presence or absence of gene sequences in these chromosomes. So we can use this in genetic researches, and this method is very widely used in oncology for detection of different oncogenes and proto-oncogenes. With this method, we can do the spectral karyotyping, in which each chromosome would be assigned with a specific probe illumination, specific color. So we can use this for studying of chromosome rearrangement, cell cycle phases, recombination of chromosome, and etc. Comparative genomic hybridization, CGH, is a technique which permits the detection of chromosomal copy number changes without the need for cell culturing. It provides a global overview of chromosomal gains and losses throughout the whole genome of tumor. Tumor DNA is labeled with a green fluorochrome, which is subsequently mixed with red labeled normal DNA and hybridized to normal human metaphase preparation. The green and red labeled DNA fragments compete for hybridization to their locus of origin on the chromosomes. The green to red fluorescent ratio measured along the chromosomal axis represents loss or gain of genetic material in the tumor at the specific locus. In addition to a fluorescent microscope, the technique requires a computer with dedicated image analysis software to perform the analysis. So students, the cytogenetic methods are the main methods of human genetics because they are used for diagnosis of different abnormalities in number and structure of chromosomes. So now these methods are used very widely, and we can diagnose without this method the human chromosomal disorders. This method we can use for diagnosis of mutation in tumors. So these cytogenetic methods can use in oncology too. But the cytogenetic method we can't use for diagnosis of gene mutation and single gene disorders. For diagnosis of single gene disorders, we can use the biochemical methods. These methods study the structure of proteins and different products of metabolism, human metabolism. So we tell which the gene causes changes in protein, and this protein can provide the different biochemical reaction of our body. So biochemical methods study the changes in protein or changes in biochemical reaction. The biochemical methods, these are chromatography. Chromatography is a physical method of separation in which the components to be separated are distributed between two phases, one of which is stationary phase, while the other, the mobile phase, moves through each in a definite direction. The chromatographic process occurs due to differences in the distribution constant of the individual sample components. There are a few types of chromatography. This is ion exchange chromatography, size exclusion chromatography, and affinity chromatography. Blotting is a technique for transferring DNA, RNA, and proteins onto a carrier so they can be separated, and often follows the use of a Gell electrophoresis. The saffron blot is used for transferring DNA. The Nathan blot is used for transferring of RNA, and the Western blot is used for proteins. So by this method, we divide the DNA, RNA, and proteins into small parts and study these parts. The next method is electrophoresis. Electrophoresis is the main biochemical technique for molecular separation. Electrophoresis can be one-dimensional or two-dimensional. One-dimensional electrophoresis is used for most routine protein and nucleic acid separations. Two-dimensional separation of proteins is used for fingerprintings, and when properly constructed can be extremely accurate in resolving all of the proteins present within a cell. We can do electrophoresis on paper, this is paper electrophoresis, but more often we do electrophoresis in polyacrylamide gel or in agarose gel. Very often we can do agarose gel electrophoresis for separation of DNA or RNA, and we separate nucleic acid into fragments of different size. So here you can see the photo of samples of nucleic acids in a gel electrophoresis, where fluorescent stain and add into samples, so we can see where. So this is picture in ultraviolet lamp. The batch of biochemical methods, these are screening tests. The screening test, these are a group of researchers which is done to detect of potential health disorders or diseases in people who don't have any symptoms of this disease. The goal of a screening test is early detection of different human disorders. Screening tests are not considered diagnostic, but are used to identify a subset of a population who should have additional testing to determine the presence or absence of disease. In screening tests, very often the different researches with low sensitivity are used. But the main feature of these researches, they are cheap, they are rapid and simple. And with help of these methods, um, we can examine a large number of people in very short time. And in these people, we found some group who have positive results, and this group of people we will examine with more expensive, more long, more severe methods with high sensitivity. The example of common screening tests, these are cholesterol measurements, fecal occult blood test, mammography, pregnancy test, prenatal screening. The prenatal screening, these are a group of methods which used maternal, placental, and fetal samples. They help to diagnose and monitor maternal condition such as gestation diabetes and preeclampsia, trophoblastic disease, and fetal chromosomal abnormalities as Down syndromes. These biochemical and hormonal tests constitute only one aspect of obstetrics care, so they should be used together with clinical findings and imaging, particularly ultrasonography. So, the screening test of pregnant women includes the examination of human chorionic gonadotropin and pregnancy-associated plasma protein A. We study these substances in maternal blood. So the studying of level of these substances help us to find the chance of trisomy 21 in fetus. But students, where less sensitivity is characterized for these tests. So If we, with help of this test, we find the trisomy 21 in fetus, we should use other more expensive, more high sensitivity test for confirming of this diagnosis. So, on based on this test, we don't diagnose this trisomy 21 in fetus. We can see us which where is the chance of trisomy 21 in fetus, but for confirming of diagnosis, we should administer amniocentesis, and in samples from amniocentesis um, or Korean biopsy sampling. So we we will study these samples and diagnosis of the presence or absence of this disease in fetus. So these tests we can use for fetus and maternal diseases during the pregnancy. And you can see a condition which we can find with help of these biochemical tests. For diagnosis of single gene disorders and human gene mutations, we can use DNA analysis. This is method which based on polymerase chain reaction. During of this reaction, the amplification of number of DNA happens, uh, and after this, we can study the sequences of DNA with help of electrophoresis or other methods. Now, is this method, DNA analysis, is very widely used for diagnosis of human complex disorders, single gene disorders. We can study with help of this method the samples from amniocentesis or carrion biopsy sampling, and diagnosis for different disorders of fetus. This method now is very used in infection disorders and microbiology. So, with help of this method, we studies the DNA of pathogen and diagnosis, and each helps in diagnosis of different disorders. The next method of human genetics is twin studies. The twin studies, this is method of medical genetic which studies role of inheritance and environment in development of different human characteristics. Where this method studies the role of environment and inheritance with help of study of monozygotic and dizygotic twins. So the twin studies is very helpful method for understanding of role of genes and environment, especially in multifactorial disorders such as body age, weight, schizophrenia, blood lipid level, and complex diseases as obesity, depression, cardiovascular diseases, and others. So, with help of this method, we can study the lifestyle risk factors such as smoking, exercise, diet. So, this method shows the influence of environment and genetics into development of characteristic. Um, in this method, we use two terms. This is concordance and discordance. Concordance, this is present of similar characteristics in twins, and discordance, this is present of different characteristics in twins. The heredity component is estimated by K.G. Holzinger. He provide the Holzinger formula where the H, this is a heredity component. This formula shows which concordance of monozygote twins minus concordance of dizygote twins. We should divide into 1 minus concordance of dizygote twins. So, as a result, we have H hereditary coefficient, and each shows the role of heredity in development of this characteristic. Um, for multifactorial diseases, where for chromosomal diseases and for monogenic diseases. So where age should be near of 100%, and it shows which were the main role in development of these characteristics where inheritance plays. For multifactorial diseases, this coefficient is between 50 and 100%. And students, as a result of these numbers, you can see the level of influence of inheritance and environment to these characteristics. So schizophrenia, yes, we see which it is 65% of this coefficient. So it means where environment and inheritance plays very important, where equal role, but where inheritance have more role in development of schizophrenia as environment. In rheumatism, we can see which the main role plays the environment. In diabetes, both environment and inheritance. The coefficient of inheritance will be less as 50% for diseases which are determined by environmental factors. These are infectional diseases as cholera, malaria. These are diseases which caused by lack of vitamins or microelements. This is rickets, goiter, and others. The next method of medical genetic. This is a population statistic method, and each studies the the genetic processes which happens in population. Gene pool with a complete set of genes of all individuals in population. Genotype frequency, this is proportion of individuals in a population with a specific genotype. Genotype frequency may differ in different population. Allele frequency, this is proportion of any specific alleles in population. For each population, the own gene pool, genotype frequency, and allele frequency is characterized. So the different population differs by gene pools. The gene frequency, the frequency of alleles and genotypes is studied by Hardy-Weinberg law. It shows which in ideal population the frequency of alleles will be constant from generation to generation. But this law is used only in ideal situation. It means so it works in ideal population, in ideal situation. So it means which in population where natural selection, migration, mutations, genetic drift should be absent. The population should be very large for free crossing. The human populations are not ideal, but we can use this law for studying of frequency of genotypes in human populations. There are three main sources sources of genetic variation in population. These are mutation. This is genetic recombination in sexual reproduction. And these are different genetic processes as genetic drift, migrations. Genetic drift and migrations. Genetic drift, these are changes in genetic pool of population due to chance, not natural selection. There are two forms of genetic drift. This is bottleneck neck effect and founder effect. The genetic drift is mostly characterized for small population. So the genetic drift, these are changes in genetic pool. In small population, many organisms are present in homozygous condition. So in small population, the genetic drift have more large influence to a gene pool. So we tell which the bottlenecked allele frequency is altered due to a population crash. So what something extreme happens, it causes the rapid decreasing the number of organisms of population, and it is not depends of natural selection. The organisms which survives give the rise to new population with a new genetic pool which differs from genetic pool of old population. Founder effect. It is very widespread in people where allele frequencies change as a result of migration of small subgroup of population. The founder effect, it is very good seen due to migration of people from Europe to America, and the people migrate to America. These people were carriers of different human disorders, and it causes the increasing of number of these recessive traits in new population, which are form which the people on which will be the people of which were ancestors were offspring of these founders. Okay, now the lecture is finished.